Welcome. How you guys doing? How's studying coming along? There's one more seat down here if you want a seat down here. And in fact, I can move that bag back there and make one more seat as well. Give people as many places to sit down as possible. Sorry. Move this out of the way. And what's that?
I was just making myself.
Okay, there's a seat here, and there's also another seat over there if you guys want. Okay, great turnout. So either that means you're very serious or you're very nervous or both. You tell me. Okay, so what I'm going to do is what I always do in these review sessions is I answer any questions that you have. As I said, I will need to leave by just a little bit after 6, and that will also help me get the video started for rendering, because that takes a while in the evening, and I've got to go meet somebody. And if I do it before I leave, then that'll be good. Okay? Anyway, the way these work is it's driven by your questions, and I am happy to answer any questions that you have. Please ask me questions specific, like don't, well, can you tell me all about da-da-da-da-da? I mean, so these are here answering questions about things that you have, problems with and so forth. If you want me to re-lecture on something, then I would say go back and re-watch the video, because that's why I have the videos there, okay? Okay, come on in, squeeze in as you can. There's one seat right here, there's one seat over there, there's one seat there, and there's a bench, and, yeah. Okay, so who wants to start? What's that?
Collagen.
Collagen, okay.
So the bonds within collagen are hydrogens and chain strands covalent. That's correct. And then
So,
vitamin C.
yeah. So collagen's a really interesting and odd compound. Collagen is actually a helix of helices. I'm not a very good artist. But there are 3 helical chains in collagen, and they themselves form a helix. All right? And so with collagen, we have modified amino acids like hydroxyproline and hydroxylysine. And these are amino acids that get hydroxyl groups put onto them after the protein has been made. So it's what we call a post-translational modification. And these side chains turn out to be interesting because if you put 2 of them very close to each other, an OH here, that's not a very good close to each other, is it? An OH, let's say here, and an OH, let's say here, okay, that there are enzymes that will actually react them and make a bond between them. Okay? Actually, that's not quite what it does. It does something like this. Okay? So, in any event, you don't even know the bond. But the point is that it's making a covalent bond where one didn't exist before, and it involves 2 hydroxyl groups. Okay? That covalent bond is actually formed as a result of action of an enzyme that requires vitamin C. So to make that bond, okay, requires vitamin C. All right? If you don't have vitamin C, you can't make that bond. And if you can't make that bond, your collagen fibers fall apart. And the analogy I gave in class was like the braided hair. If it doesn't have a tie on the end that holds it together, it will fall apart. Okay? And so that's what's happening with collagen. Okay? Yeah?
Is that bond the covalent bond? Or is that
That's right. This is a covalent bond right here. That's a covalent bond where one didn't exist before. So, and what happens is a bunch of these bundle together. So you'll have bonds within a helical chain like this. You'll have bonds between chains. So you have a bond like that. Okay? And so these bonds give structural integrity. It's kind of like, again, braiding something gives you more strength than the individual components of the braid. This helps hold the braid together. And so the more bundled these things are, the stronger the collagen is. And that's why if you're deficient in vitamin C, your collagen bundles don't stay together. And as a consequence, the collagen literally falls apart. Since the collagen is the glue that's holding you together, you fall apart. That's the nasty side of it. So it's rare that anybody has that anymore. There's so much vitamin C in our diet, so much vitamin C and fruits and so forth available, that to get scurvy, you really had to literally eat a salted pork diet for months on the ocean to get it. All right? It doesn't take much vitamin C to make these sorts of bonds. Yeah. So it's a good question. So what happens is you have two OHs that are reacting, and so what you do is you actually split out H2O. That's why I drew it wrong originally. All that's left is that. So water is splitting out, and you're left with that. You don't need to know that. I'm just showing you that for your own information. But that's what's happening with the putting the two of them together. Yeah.
Is there anything else you want us to know about those two that the hydroxyl and hydroxylisin? Can you talk about how they're usual interactions, but develop?
Sure. Well, anything I talked about is fair game. So that's always the answer. So if I talked about it, it's fair game. Yeah. That's not a hydrogen bond. That's a covalent bond. That's a covalent bond on each side. You notice there's no hydrogens involved. So that's right. That's a covalent bond. And covalent bonds are strong. That's what you need to make your collagen hold together and be strong.
Are the OH groups put on are the OH groups that hold the separate strands put together at the same time with the OH groups that hold? Sorry. Are the OH groups that hold the three separate strands together put on at the same time as the ones that form the helical shape and individual strands? Are they all hydroxylated at the same time?
So I'm not sure I understand the question, but all the hydroxylation happens as enzymes are passing through and doing that. So yes, I'm not sure what you mean by.
What keeps the carboxyl groups that eventually make that triple helix from just one to more convoluted helix in one individual strand?
What keeps this from becoming less helical?
So why do the carboxyl groups that eventually end up holding multiple?
There's no carboxyl groups I'm showing here.
Sorry, the OH groups that eventually hold multiple collagen fibers together.
Uh-huh.
Why don't they interact on the strand that they're actually on?
They may. That's why I drew it here, and I also drew it here.
Is there a protein that's like chaperone that helps?
So there's an enzyme that catalyzes this reaction. It's this enzyme that catalyzes this reaction that's making that possible. And that's the enzyme that requires vitamin C. Okay? Yes? Okay, so that's a good place to come back to it. Okay, okay, I'll talk about it. Anything more on this before I move to that? Okay, I'm going to move it over here to split up the stuff. How's that? So, understanding buffers is really critical, okay? Because without buffers, we're not alive. We don't have anything that takes care of those protons that get produced by metabolism and so forth. So we really need buffers. What you're referring to is this equation that I wrote to start discussion of buffers. Okay? And I've talked to about 5 or 6 people now, and every single person, when I sit down with them, they're not starting with this equation. This equation reminds you that what's happening in a buffer solution. It reminds you that there is a weak acid. And it reminds you that there is a salt. And the only difference between a weak acid and a salt is a proton. That's the only difference. Okay? Now, if I take a salt and I add a proton to it, what do I get? I get a weak acid. And if I take a weak acid and I take a proton away, I get a salt, right? All right, so that's what's happening in a buffer. If I add protons, all right, then I'm putting protons in here. The protons don't just sit around and do nothing, because the buffer is resisting a change in pH. It's going to try to keep the impact of those protons from affecting the pH very much. So if I add protons, something's going to try to take them, and there's only one thing here that can absorb protons. What is it? Salt. Salt is going to absorb protons. How many does it absorb? Well, if I had, let's say, 100,000 of these, and I had 100,000 of these, and I added 10,000 protons, what would happen? Well, 10,000 of these would grab one proton each, and I would end up with 90,000 left, because I would lose 10,000, and I would create, in the process of that, 110,000. So all that a strong acid and a strong base is going to do is interchange these two. If I add a strong acid, this gobbles it up. When it gets gobbled up, it doesn't exist anymore, right? You put a proton on here, it no longer is a salt, it is now a weak acid. If I put sodium hydroxide in there, the opposite happens, because this thing gobbles up protons. So let's say I had this mixture, and then I added, let's say, 40,000 hydroxides. What am I going to get? Well, what I'm going to get is 40,000 protons being taken away. This is going to try to replace 40,000 protons. So 40,000 of these guys are going to give up protons. When they give up protons, they're not HA anymore, right? So I have 100,000 minus 40,000, or a total of 70,000 left. Okay? And I add 40,000 over here, and I get 130,000. So the point is that for every manipulation I do with a proton, I either make acid or lose acid, make salt or lose salt. And they're interchanged. That's what a strong acid and a weak acid are doing. Okay? Does that help? Okay. Very important. Yeah?
You say they're at max capacity, buffers at maximum capacity.
So when I say a buffer is at maximum capacity, what do I mean? All right? So on a graph, I'm not even sure if this is showing up on the thing. I should probably be careful about it. On a graph, okay, if I have pH versus OH added, all right, I saw that graph do something like this for an amino acid, right? And I said, well, this was pKa number 1. This was pKa number 2, right? Everybody agree with that? Everybody remember that? Okay. So your question is, what does maximum buffering capacity mean? Well, maximum buffering capacity means the maximum ability of the buffer to resist a change in pH. The maximum ability of a buffer to resist a change in pH. Well, when will that occur? That will occur when you have maximum amounts of both of these. So when I have equal amounts, 100,000 and 100,000, that is when my maximum ability to resist change in pH is, because this guy can grab 100,000, this guy can donate 100,000. That's the best place it's going to resist the change in pH. So when these two guys are equal, that's maximum buffering capacity. Well, it turns out if you do the Henderson-Hasselbalch equation, you can solve that yourself. You see that pH equals pKa plus log of S over A. And when S equals A, the log term equals 0, which means that maximum buffering capacity equals when the pH equals the pKa. And not surprisingly, that's where the graph is the flattest. Flatness means it's not changing in pH as we're adding more OH. Does that make sense? Okay. Yes? Yeah, so what if you add salt to a weak acid buffer? All right? Salt's not protons. Salt's not hydroxyl. And that's what's causing that interchange. I'm just adding more salt. If I add more salt, I'm actually making more things there for the buffer to have, right? So if I added, let's say, 20,000 salts to this, what would happen is I would have 120,000, and I wouldn't change anything there because I haven't done anything to change protons, right? So addition of salt would do exactly that. It would just simply add to it. Okay? Yes?
What holds together
What holds together quaternary
quaternary structure?
structure? It's a good question. You know, I'm not even sure if I said that, but I'll tell you here. Since I didn't say it, I won't hold you responsible for it. But I'll tell you, you should want to know this, right? So we're here to learn things. So it turns out that the very same forces that stabilize tertiary structure stabilize quaternary structure. That includes the hydrophobic interactions, ionic bonds, hydrogen bonds, metallic bonds, and disulfide bonds. All of those will help to stabilize quaternary, just like they help to stabilize tertiary. Okay? Yes?
Question about collagen. What level of structure is that compared to three different subunits, usually fibrous,
Yeah, it's a good question. So collagen is considered a fibrous protein because it's really not bent. It's really not folded up in any sense. These are links between these adjacent ones, but the real protein is really this part right here. So we consider it to be purely a fibrous protein. And I agree. One of the things I've had a couple questions on, and your question is consistent with that too, is that there are things that people look at me and say, well, that's not exactly secondary structures. When I talked about beta sheets, for example, beta sheets were made by sort of interactions between beta strands. And I said, for the most part, that is, in fact, a tertiary structure. And I said, well, the book says that it is a secondary structure. It depends on where you read about this. But in fact, beta sheets, okay, are an interaction that make a tertiary structure. Beta strands are a secondary structure. So if you go on Wikipedia and you look in a variety of places, you'll see confusing terms with that. Yes?
So in the highlights it says that in the lungs is acidic, and so that forces the carbon dioxide off, but in acidic environments, I thought oxygen had trouble to be picked up by hemoglobin.
Okay. So that line I sort of wish I hadn't put in the highlights because I've had a lot of people concerned and confused about it. So her question had to do with, there's a line in the highlights that says the environment is more acidic and so carbon dioxide is released, and doesn't acidic conditions cause hemoglobin not to want to bind oxygen? Okay, so a couple things. One, the localization of the acidity is really, it's not like I'm saying all the lungs are that way. Okay, so it's really fairly localized where this exchange is happening, and so that kind of confuses people, number one. Number two, just because you're in an area where it's slightly more acidic doesn't mean it can't bind oxygen. It means it might be more prone to let go of oxygen. In the lungs, we've got a very different situation than we've got in the rest of the body. The lungs, the oxygen concentration is enormous. It's really high compared to the rest of the body. So even though we have situations where we don't have sometimes an ideal binding environment, when you bring hemoglobin to the lungs for the first time, hemoglobin's in the T state. It doesn't want to bind oxygen at all. It's that first one that binds that causes the flip that causes the cooperativity to happen. So the lungs are not set up as an ideal environment to bind oxygen. But because there's so much oxygen there and this phenomenon of cooperativity happens, that they get pretty well bound up with oxygen. So the pH difference that's there isn't significant enough to be a problem. Okay.
Is there another reason you don't see CO2 is because there's low concentration CO2 there?
Well, the acidity plays an important role, actually. So the acidity actually comes from, the reason I say it's localized is the hemoglobin's carrying protons. So it's dumping some protons right there. You got protons right there ready to help make this carbonic acid. With carbonic acid, if you look at the reaction that I showed you, carbonic acid forms if you take water plus carbon dioxide and you go to the right, you make carbonic acid. But if you take carbonic acid and you go back the other direction, you make carbon dioxide and water. So it's reversing the reaction. So it's partly a concentration phenomenon as you're talking about, but it's also partly an important consideration for acidity. Yeah. Yes? Can a molecule bind 2,3-BPG and CO2 at the same time? The answer is yes, it can. Because remember that CO2 is binding and other places in the hemoglobin molecule. And the only place 2,3-BPG is binding is in the donut. So they're not competing in any way.
Can you explain
Good
the
questions.
difference between 2D gel electrophoresis and regular?
Yeah. 2D gel electrophoresis versus regular gel electrophoresis. Okay, I'm going to go and do this here. So, okay, so 2D gel electrophoresis. 2D refers to 2 dimensions. And if you want to think about it meaning 2 different gel techniques, that's fine too. Okay? So 2D gel electrophoresis relies on the 2 techniques being done in the right order, and the 2 techniques obviously doing their own respective contribution to the overall product. I think 2D gel electrophoresis is pretty cool. If you look at 2D gel electrophoresis, it relies on a technique called isoelectric focusing. Isoelectric focusing is done literally in a tube. Okay? And the tube has in it a gel that will basically have ions that will separate according to charge. If you put your protein in this mixture with this gel, the proteins themselves also will separate according to charge. So you make that happen by applying electricity to it. So you put a positive charge over here, you put a negative charge over here. That means that on the inside, all the negatives are going to line up over here. All the positives are going to line up over here. The most positive ones will be closest. The most negative ones will be closest. In here, we get some more others. Okay, so this might be plus 10, 11, 9, 8, 7, something like that. And the middle, we're going to have 0. So isoelectric focusing allows us to basically separate molecules on the basis of charge. That's what we're doing in isoelectric focusing. We're separating molecules on the basis of charge. So that's the first D, the first dimension. That's the first thing that we're doing in that technique. Then we literally open up the tube and we put it on the top of a polyacrylamide gel. When we put it on the top, we also add SDS. Why do we add SDS after and not before? It'd be a really good exam question. So if we put it on before, everybody would be negative, right? And we'd all go one direction. So we wouldn't be separating on the basis of their charge. Why do we have to add it at all? Because we want to separate the proteins based on size, okay? And to do that, we have to have, remember, up here is going to be a negative electrode, and down here is going to be a positive. So everything is going to get pushed through and pulled on the basis of that. So if these guys aren't all negatively charged, we're not going to separate them on the basis of size. Okay? Because what's going to happen is, well, the positives are just going to go shooting up there. They're not even going to enter the gel. So we have to add the SDS to make that happen. And so we do that. And so when that happens, then, we find that we get a variety of spots. You get thousands of spots on an individual gel. And you can look at this and you can say, oh, well, remember that in this dimension, polyacrylamide gel electrophoresis, we are separating the basis of size, so we get large at the top, large at the top, small at the bottom. And so now we can actually say this guy was a medium-sized one that started out very positive. Here was a large one that started out roughly 0. So we've actually characterized the proteins in this technique. Now the beauty of the technique is that if you can compare one gel to another. So as I said in class, if you took the proteins from a cancer cell and you did this separation and you compared it to the profile you got from a normal cell, what you would see is over here in the normal, you might see many of the proteins are going to line up exactly the same, but you're going to have some that are going to be different. And you're really interested in, well, what is it that makes a cell cancerous? Is it a new protein that's being made, or is it a protein that's not being made? Maybe it disappears. Okay? So that comparison really tells us an awful lot of information about the difference in those cells that we're comparing. That's what 2D gel electrophoresis is most useful for, actually. Yes? That's right. So for a 2D gel it's a good question. For a 2D gel electrophoresis, they typically do not run a ladder because the ladder it would have to be in 2 dimensions. And so you don't want to contaminate the other things that you've got in here. For what you're talking about, a ladder, what she's talking about is for a one-dimensional, just a polyacrylamide gel electrophoresis, where you're separating in one dimension, you'll see things come down as a variety of bands. Okay? And a ladder is a set of proteins of known sizes. So you could look at this and say, oh, well, this is 80,000 molecular weight, so this guy over here must be about 90,000 in molecular weight. The ladder tells us that. We don't use something for this. But it's not really much of a problem because we can characterize, we know the positions of the proteins of cells in a 2D gel. So we have a pretty good idea of what that is. We don't really need the ladder quite as much. Yeah, so is acrylamide like agarose? In principle, yes. Agarose I like to think of as a sort of a mesh. Okay, the mesh is just a set of structures like a sponge. Imagine a sponge. Sponge has holes in it, right? So the agarose is like a sponge, and the only difference between the agarose sponge and a polyacrylamide sponge is the size of the holes that's in it. So the holes are where the molecules are moving through during electrophoresis. And an agarose gel has big holes, great big holes, because the DNA itself is quite large. And if we don't have the holes big enough, the DNA isn't even going to enter the gel. Okay? For proteins, which are much smaller, we have to have smaller size holes, and that's why the polyacrylamide is important. Okay? How are you guys feeling about the exam? Are you anxious, nervous? What are you thinking? You can hear the, oh. I'm guessing that's anxiety. So as I've talked to several of you in my class, I think anxiety and just worrying about things are really bad things for performance. I really wish I could get people past that anxiety mode. It's really important to just use the heads that you've got. You've got very good heads on your shoulders. You don't get to this point if you don't have that. So relax and do well. One year I decided to make basically an exam where, and maybe you've seen some of the questions from the old exams where I had various people doing various roles in the thing and so forth. And I thought this would be really fun. People would relax and do it. And I talked to people who got so stressed by it. And it's like, oh, well, okay, I guess it kind of defeated that purpose. So I didn't do that this time. Yeah.
So for 1D electrophoresis, is it more on
well, time is a factor because you don't want to run it too far. Because if you run it too far, then they start coming off the bottom. So yes, in a sense, that's true. But the aim here is separating on the basis of size. So what you'll do when you run a 1D, for example, is you'll run a colored marker that'll tell you, and it has a known size. And so the colored marker, when it gets down here, across the thing, you're going to say, okay, we're going to stop it right there. That's typically what people will do. Sure. Yes. But they are single bonds. That's right. Yep, you got it. And the reason they act like double bonds is because of this phenomenon I talked about called resonance, where it basically borrows electrons from the carbonyl oxygen. And so that leaves the carbonyl oxygen in a single bond state, and the electrons pop down into the carbon and nitrogen bond. Yeah.
When you talk about the complexity of protein folding and how it takes forever for
Yes.
a million to do
Literally forever, yeah.
does that have anything to do with like each individual sort of chromosomal slots and like each of those angles that it can be between each
Yeah.
amino acid, or why?
Yeah. Okay, so our question is, when I said it took so infinitely long to fold a protein on a random basis, Did that arise from the various possible angles that are present in Ramachandran? I think that's what you're saying. The answer is basically yes. Basically yes. We think about the rotations that can happen there, okay, across that alpha carbon. Those rotations are 2 of the degrees of freedom that a protein has. So those rotations will affect the polypeptide backbone. The others will affect the R group. So the R group has got all kinds of single bonds in it. So the R group can have all kinds of different configurations with that. And what happens is the way a protein actually folds, we think, is that as it's being made, those things start interacting with each other. And those interactions, one interaction defines what that first structure is going to be, which defines what the next structure is going to be, which defines what the next one's going to be. And so as I told some of you who asked me after one of the lectures, the reason that you typically can't take a protein, boil it, and have it come back together is that those folds start in a sequential manner. You didn't have everything present. So it's too confused to try to come back together if the whole protein is there. Because they actually literally had to start folding without the carboxy terminus being there, for example. But when the carboxy terminus is there, it gets in the way. And so most proteins will not refold properly as a result of that.
Is it just the carboxy terminus, or is it all those other R groups further down the cell?
Well, by the carboxy terminus, I'm talking about everything beyond the first couple of amino acids. Okay? So, when you start folding, you can start literally folding, you can start making secondary structure, for example, with the first 10 amino acids. Okay? So if you have the first 10 amino acids and it's nicely making this little alpha helix that it starts making as it's being synthesized, there's nothing that's there to bother it from later, right? But what if you now have the whole protein that's there and you boil it? Well, you get randomness that's here. The carboxylase down here all of a sudden decides, oh, wow, I like this amine group on amino acid number 3. Well, it interacts there. Now that amino acid number 3 is not going to be part of an alpha helix. You see, so I'm trying to keep it simple, but that's the complicated kind of thing that happened once you unfold a protein. So it's rare that a protein will come back together properly after you've boiled it. Very rare. RNase is one. Yes. Oh, okay. So how do excess protons denature a protein? All right, let's imagine, if we will, I'm going to use my artistic ability to draw a protein for you. And I've got a protein that has a structure like that. Okay? And that structure arose as a result of the folding that happened during the synthesis of that protein. Okay? We can see that this has several places where we might expect we have some interaction. So we certainly have within secondary structure, we have hydrogen bonds that are happening between the individual units. We have hydrogen bonds that are happening here. We might even have some hydrogen bonds that are happening back and forth here. Okay? But remember, the hydrogen bonds are not the only thing that affect the structure of a protein. We had disulfide bonds, hydrogen bonds, hydrophobic bonds, and ionic bonds. Ionic bonds are actually the answer to your question. So ionic bonds are helping to stabilize a protein. So we might have, for example, a COO minus right here, and we might have an NH3 plus right here, and now there's an ionic bond between them because they're attracted to each other. That can be present in many places in this protein, but there's one right there. Okay? Well, if I change the pH of the solution, and what you guys have been seeing happens with buffers, what happens? Well, if I start adding protons, things can start grabbing them, right? Well, this guy's already full of protons. It can't get any more, but this guy can get a proton. This is a salt. So if I put a proton on there, what's going to happen? Well, instead of being a COO minus, it's now going to be a COOH. The force that was holding these two together is no longer holding them together. This guy now can just go flopping around. Well, multiply that times 20 or 30 within a protein, and the integrity of the protein is going to change because what was fairly tightly held together is now loose. okay? And it falls apart. Does that answer your question? Yeah, good question. How is 2,3-BPG removed from hemoglobin? Okay. The answer to the question is that 2,3-BPG, like oxygen, is not bound covalently. Okay. So anything that is not bound covalently has what I call an on-off relationship with the molecule, with the thing that it's attached to. Okay. It's being held in place, but it's not covalently bound. Covalent bonds, you've got to lock it there. You're not going to break those covalent bonds easily. But something that's just held in place by charges or whatever are not stably held together. Well, that's how 2,3-BPG binds. Okay? So it means that 2,3-BPG can get in the middle of that hemoglobin molecule, and it can come out. It can go in. It can come. That's the on-off relationship. Okay? And so when we see things that bind non-covalently, that's what's happening with them. They're on-off. They're on-off. If 2,3-BPG, as it's making its way back to the lungs after it's bound to the hemoglobin, comes off and a cell grabs it, then it doesn't make it back to the lungs. And that's actually what happens. So on the way back to the lungs, the on-off relationship, there's billions of cells that it's encountering along the way. One of the cells just grabs the 2,3-BPG. And 2,3-BPG is actually useful to cells as an energy source. And we'll talk about it when we talk about glycolysis. But it's useful to cells. They'll take it and they'll use it for energy. And so they're doing the rest of the body a favor in the process of doing that, because as the hemoglobin gets back to the lungs, now it doesn't have 2,3-BPG. Okay? Yeah? Okay. So why is it that smokers have a harder time, is what you're asking me, I think, right? Okay. So smokers have higher levels of 2,3-BPG in their bloodstream. Okay? Now you've all got some 2,3-BPG in your bloodstream, because it's coming on, it's coming off, it's falling off. Sometimes it's grabbed by a cell, sometimes it's not. You all have a little bit of 2,3-BPG in your bloodstream. The more 2,3-BPG you have in your bloodstream, the more likely it's going to make it back into the lungs attached to hemoglobin. Now that's important, because when hemoglobin makes it back to the lungs and it's attached to 2,3-BPG, what did 2,3-BPG do to hemoglobin? Why is it that it's letting go of oxygen? It's put in the T state. And the T state is a state where it doesn't like to bind oxygen. So if the hemoglobin makes it back to the lungs with 2,3-BPG, that hemoglobin is going to sit there in the T state. It's not going to bind hardly any oxygen at all, if any. So the higher your levels of 2,3-BPG are, the more likely that's going to happen. So what it means is that as a percentage of all your hemoglobin, the carrying capacity for oxygen is decreased in a smoker, because they have more of it in the T state in the lungs than they have that can be in the R state to bind oxygen. All right? A non-smoker has a much lower percentage of hemoglobin that doesn't bind oxygen. So they've got a lot more oxygen carrying capacity. Does that make sense? Very good question. Is oxygen bound in the same way that 2,3-BPG is? Oxygen turns out to be bound a little bit more tightly than that, but it's not a strong covalent bond, no. So and that's good, because you want oxygen to come off. You do want it to come off. As oxygen is just traveling on its route, some of it's going to come off. That's going to happen. And that's good, because again, we want to have all of our cells have access to oxygen. Not all of our cells are making 2,3-BPG. And not all of our cells are producing protons because they're metabolizing rapidly. So we do want some oxygen just to be dumped off, and that does happen to some extent, yes. Yes, sir. So 2,3-BPG binds in the donut hole, and the oxygen binds in the porphyrin ring. That's correct. But it's the structure of the protein that's being affected, which is what's affecting the oxygen binding in the porphyrin ring. So even though that they're not interfering with each other, the structure, it's an indirect phenomenon. Yeah.
But even when so does the hemoglobin lose the BPG and then it gets back to the lungs, but it's still in the T cell?
That's right. Very good observation. You're exactly right. Did you hear what she said? So once hemoglobin gets flipped into the T state by 2,3-BPG, it stays in the T state. It's going to go back to the lungs. It's sitting there in the T state when it gets back to the lungs. That's why cooperativity is so important. Remember, again, we've got a very high concentration of oxygen in the lungs. We literally have one oxygen that forces its way onto a hemoglobin, and the whole hemoglobin flips now so that it wants to bind oxygen. Okay. I don't want to leave the impression that T state it won't bind any oxygen. It's just reduced binding affinity for oxygen. That's important to understand. Okay. And that's why that high concentration of oxygen in the lungs is important. It's going to force itself on there and then make all the other bindings possible. But yes, it'll be in the T state when it gets back to the lungs. Indeed. Yes?
When we talked about the percent saturation curve,
Yes?
and hemoglobin being sigmoidal, can you explain why sigmoidal versus low?
Okay, so why is the oxygen binding curve for hemoglobin sigmoidal instead of being hyperbolic, is what we said, right? Okay. So it turns out it's because of the phenomenon of cooperativity, and the sigmoidal plot is actually a manifestation of that. Okay. So a plot, oxygen binding plot, where we have percent saturated versus concentration of oxygen that's here. If we compare myoglobin with the plot that we see for hemoglobin, myoglobin is hyperbolic, hemoglobin is sigmoidal. Okay. The reason that this is the case is that if you think about it, hemoglobin exists in two states. It exists in a T state and it exists in an R state. All right? In the T state, hemoglobin is less likely to bind oxygen. In the R state, it's more likely to bind oxygen. Right? Okay. So R state up here, T state down here. And so once we force oxygen onto hemoglobin, it starts, oh yeah, I want to grab some, and it moves into the R state. That's what you're actually seeing in this plot. Mm-hmm. Yeah. Okay, so that'd be a good exam question. So I save that for it. Where would fetal hemoglobin be? Okay. Well, let's think about it. You guys can figure this out. We'll do it. Fetal hemoglobin is a sigmoidal plot also. Okay. All right. Now, fetal hemoglobin does go in the T state. It just doesn't get forced in the T state by the 2,3-BPG. Okay? That's important to remember. It's not important for us. I haven't said that in class, so don't worry about that. But I'm just saying in the overall. When I say something's important to remember, everybody goes, write this down because it's going to be on the exam. Right? While exams are important, okay, and I really want you to do well on your exams and everything, that there's more important things than exams. I know that's hard to believe, but that is true. Okay, there are more important things than exams. All right. So anyway, so if we think about fetal hemoglobin, what were the characteristics of fetal hemoglobin? It took oxygen away from mom, right? Had to have a greater affinity, had to be able to hold on to oxygen better than mom's did, right? Which of these two had greater affinity, myoglobin or hemoglobin? Myoglobin, right? Because even under lower oxygen concentrations, it had a pretty fair percentage bound. So if I told you that fetal hemoglobin was sigmoidal in nature and it had a greater affinity, what's it going to look like? It's going to be above it. It's going to look like this. Right? Because it's going to, at the same, so look at this, at the same oxygen concentration, here's mom, here's the fetus. Mom has, let's say, 30%. The fetus has 40%. The fetus, under the same oxygen concentration, is holding on to more oxygen. That's greater affinity. That's what that means. Okay? So, a question about mom being a smoker, that doesn't affect the fetus. That's not true. Mom being a smoker does affect the fetus, but the 2,3-BPG doesn't affect the fetus. Okay? The fetus is definitely affected by mom being a smoker. Don't walk away from here with the impression there's no effect. Okay? Please. Okay? I'm very anti-smoking, so I don't want to leave you that impression. I may have left you that impression. I don't want to leave you that impression. The fetus is affected because mom's oxygen carrying capacity is reduced. If you look at the birth weights of the children of smokers and compare them to non-smokers, there's a difference, and it's because the fetus is starving for oxygen while mom's smoking. Okay? It's just that the 2,3-BPG doesn't affect anything. That's the only message there. Yes? That's right. That's correct. But if I put 2,3-BPG on here and I did the same sort of thing, here's what you could predict what you would see. what would you see? What would the plot look like? It would be more down here, right? That's what it's going to look like. So again, graphs aren't just squiggles that tell us nothing. Graphs are telling us something, and we can understand things by looking what is on a graph. Okay, that's really important to that is important to understand. Okay, yes. This one right here, yes. Not picking up as much, not getting as much. Good. These are good questions, guys. I've never had a 350 class that came in such large numbers to a review session. This is very cool. Maybe it's not cool. Maybe you really are paranoid or something here. I don't know. I hope it's because that you're really interested in this and that you're all going to do well. That would make me feel very good. I really would. What's that? Now you have the pressure is on, right? So the exam is written, and of course I can't tell you anything about the exam, but I can say that I always strive to be fair in an exam. I don't believe in trying to trick people and things like that, and so I will always challenge you on an exam because what I like to see is you thinking. Thinking is important, and but I also think it's a fairly straightforward exam. At least I hope you think that as well. There are 3 sections on the exam, and the format is almost exactly what you saw in the practice. In fact, it is exact. I shouldn't say almost. I can't remember. The points might be slightly different or something, but other than a point or two here or there that's slightly different, there's really no change in the format of the exam. Okay? So you've got a section that is the multiple possibilities, and you notice that that's not a multiple choice. You're basically marking whether each one's true or false is what you're doing on the that first section. The second section is short answer, and the third section are longer answer, and that's the format I always use in this class. Yes. Okay. Yeah. So the difference between gel electrophoresis and gel filtration. So gel filtration is the technique I described that uses the tiny beads with the tunnels through them. And we're separating molecules on the basis of size. In gel exclusion, or gel filtration, same thing, the smallest molecules travel the slowest because they go through all those little tunnels that the big molecules can't get in. So the big molecules come out first, the small molecules come out last, and we're separating on the basis of size. Now the only parallel to the gel electrophoresis is that we're also separating on the basis of size, but the means of separation is completely different. So in the case of gel electrophoresis, we have this mesh, kind of like this sponge that I talked about, through which the molecules are being pushed by an electrical field, electrical current. Okay? And that electrical current is the driving force. In the case of gel exclusion, we simply have just diffusion. The molecules are just moving on their own rate through there. So there's not really a good relationship between them. And then we also remember with gel electrophoresis, it's the opposite. Okay? We have the smallest molecules moving the fastest because they can navigate that sponge faster than the large molecules which get trapped as they go further down through the sponge. Okay?
When you're talking about enzyme inhibition,
Yes?
you said that drugs are developed as competitive inhibitors.
Commonly, yes. Uh-huh.
Is there a reason that they're competitive instead of non-competitive?
Yeah, it's a very common question. In fact, I think you're maybe the second or third person that's asked me that question since I gave a lecture. This is a good question. So the question is, why is it that we don't make drugs that are non-competitive? I didn't say that we don't, but they're harder to do. And the reason that they're harder to do is when you make a competitive inhibitor, you know what the active site looks like. You know, because you've already got the molecule that binds to the active site, you're just mimicking it. Now, if you're making a non-competitive inhibitor, you're not binding to the active site. You're binding to another site, and you're not even sure what that site's going to look like. And if it does bind, does it affect the enzyme in a way that is going to cause the enzyme to not be able to work? So you have two variables when you do a non-competitive compared to a competitive.
Couldn't you compare it to another non-competitive inhibitor that you know?
That would mean you had to know of one.
Is there always?
So there's not. I mean, so, people spend, drug companies spend a lot of time investigating inhibitors of any type, whether they're competitive or non-competitive. And again, I'm not saying that there aren't non-competitive inhibitors that are out there. I'm just saying that it's easier to design a competitive one, much easier.
Could you maybe go over the effects of a competitive inhibitor?
Sure. Okay. So competitive inhibitor versus a non-competitive inhibitor. All right. Let's think about what's happening. Competitive inhibitor. Describe a competitive inhibitor to me.
It binds
It binds the active site, which means it has to look like the substrate. Okay? So competitive inhibitors look like the substrate, and that's where they get the competitive name, right? Competitive means that they're both competing for the same site, right? Okay? Well, they're competing against the substrate for the enzyme, right? The enzyme we're trying to stop an enzyme from acting, right? And we stop an enzyme from acting by stopping its ability to normally catalyze a reaction on the normal substrate, right? So a competitive inhibitor is going to compete with that normal substrate for the active site. That's what a competitive inhibitor is going to do, right? Well, if they're both going to bind to the if we're going to compete, it's going to have to bind to the active site, because if it can't bind to the active site, then it's not going to be a competitive inhibitor, right? All right, so by definition, a competitive inhibitor will look like the substrate and will bind to the active site. If I look at V versus S, velocity versus substrate concentration, for an enzyme that has no inhibition, I'll see a sigmoid. I'll see a hyperbolic plot like you see here. Okay. If I do the same reaction in the presence of a competitive inhibitor, all right, what's going to happen? Well, what's going to happen? You might think, well, why do we use a competitive inhibitor? Because the substrate's going to win, right? Well, the substrate's not going to win in the body. This is in a test tube. We're doing this in a test tube. So in a test tube, we're doing a series of maybe 10 different test tubes. All right? The test tubes all have the same amount of enzyme. They all have the same amount of inhibitor. That's key to what you're seeing here. Notice what I said, the same amount of inhibitor. And they have varying amounts of substrate. Each tube has its own characteristic amount. That's what's going out here, right? I may get out here to 1,000,000 to 1. That is 1,000,000. It's 10 million. Okay. No, it's 1,000,000. 1,000,000 to 1 of substrate to inhibitor. If I did that in my body, the inhibitor would have no effect. So I have to give in the body, I have to give enough drug that the inhibitor is going to compete effectively. It's going to be more down in here. Well, down in here, what's the velocity of the enzyme compared to the enzyme normally? Right? It's way lower. It's way lower. Which means, therefore, that the drug is knocking the enzyme down, the body has a chance to overcome the thing that's happening. Now, to get back to the point, remember that we've got varying amounts of substrate. Down here we've got low amounts of substrate. The ratio of substrate to inhibitor is low. The inhibitor is very able to compete. Up here, the ratio of substrate to inhibitor is high, because remember, I have the same amount of substrate, same amount of inhibitor in each case, but increasing amounts of substrate. So what's it going to look like? Well, when I start out, it's going to be very low. I'm going to get very little velocity, or at least reduced velocity, because the inhibitor is effectively competing, and the enzyme can't make product because it can't bind to substrate. But as the substrate concentration gets higher and higher and higher, then the substrate starts flushing out the inhibitor. It's competing because it's got a numbers advantage, maybe 1,000,000 to 1. Okay? That numbers advantage means that the enzyme effectively doesn't see the inhibitor. It's going to be 1,000,000 times more likely to bind to the enzyme, to the substrate, than it's going to be to bind to the inhibitor. I will never see the inhibitor. 1,000,000 to 1's a pretty good size ratio. Okay? So when that happens in a competitive inhibition, if Vmax is here, all right, Vmax is essentially the same for the two. I don't see a change in Vmax because at high concentrations of substrate, the enzyme doesn't even hardly see the inhibitor. Okay? What happens to Km? Well, if I take Km, so I did this for somebody earlier today, and they said, well, how did I decide where Vmax over 2 was? Well, Vmax over 2 is going to be half the way up here, right? It's going to be half the way to Vmax. There's Vmax over 2. Remember that Km is the substrate concentration that gives Vmax over 2. It is not Vmax over 2. It is the substrate concentration that gives Vmax over 2. Big difference. Okay? Big difference between the two. All right. So what happens to Km? We're talking about competitive inhibition now. What happens to Km? Well, here is the where the Vmax intersects the curve. Here is the substrate concentration that gives Vmax over 2. That is the Km right there. That is a substrate concentration. Km is a substrate concentration. Okay. Well, what is it for the inhibited reaction? Right. Well, for the inhibited reaction, Vmax over 2 intersects right there. We look at the substrate concentration for that, and that is the Km for with the inhibitor present. The Km has increased. All right? The Km has increased in the presence of the inhibitor. What if we did the same thing and we had a non-competitive inhibitor? Well, a non-competitive inhibitor, all right, means that, I gave the example in class, that if I had 10,000 molecules of enzyme and I had 5,000 molecules of inhibitor in a non-competitive reaction, 5,000 enzymes are always going to be bound to the non-competitive inhibitor. It's not going to matter how much substrate I add. And the reason it's not going to matter is because they don't compete. There's no competition whatsoever. Once the inhibitor has bound, that's it. Okay? They don't look alike. I can add all the substrate I want, but it's still not going to plug up the binding site for the non-competitive inhibitor. The non-competitive inhibitor is going to bind to a different site. It's always going to do its own thing. So, what that means is that for non-competitive inhibition, we effectively always have a reduced amount of enzyme compared to an uninhibited reaction. We always have a reduced amount of enzyme, and it's a fixed amount. It's a fixed amount. So if I said to you, I'm going to have to finish up here in a few minutes. All right. If I said to you, all right, I'm going to do 2 reactions. Forget the inhibitors. Here's a reaction, I put 10,000 molecules of enzyme. Here's a reaction, I put 5,000 molecules of enzyme. Am I going to get the same Vmax of those 2? No, I'm not. Because Vmax depends on how much enzyme I use, right? 10,000 is going to give me twice as high of a Vmax as 5,000 is going to give me, right? Well, now I do it with an inhibitor. With an inhibitor, I've still got 5,000 active molecules of enzyme compared to 10,000 molecules of enzyme. I know, therefore, that Vmax is going to decrease when I have a non-competitive inhibitor. It's going to look like this. That's what it's going to look like. It's going to have a new Vmax. Okay? That right there is going to be Vmax for a non-competitive inhibitor. A different Vmax because I've got a reduced amount of enzyme. What happens to the Km? Okay. Well, if that's the Vmax, then let's say that the Vmax over 2 is about right there for the non-competitive. All right? If I go over here, what do I discover? I have the same Km as the uninhibited reaction. That's the second time today I've drawn that thing accurate enough that it turned out good. All right. Now, it's not just artistic ability that makes that be the case. There's actually some very important reasons why the Km does not change. But suffice it to say, for our purposes, you simply need to know the Km does not change for a non-competitive inhibitor. Well, Vmax over 2 changes because the Vmax changes, right? So Vmax is different. Vmax over 2 is going to be different. That's what this is why I put NC here, non-competitive. Okay. Very pleased with the questions here. This is very good, guys. I'm not chasing you off. I'll take another 1 or 2 if you have any others. Everybody's ready for dinner. Okay. Good luck. Study hard. See you tomorrow.